introduction, and thank y'all for being here. So I'm going to talk to you about my project, which is about the mechanisms of weed suppression by a cereal-like cover crop. So why should we research cover crops? Well, one of the big things, this is one of the easiest, cheapest ways of doing erosion control. And like, you've probably heard this in soils intro class at some point, and I've heard that it takes about 100 years to form 1 inch of soil, and about 1 rainy year to lose that inch if you leave your soil unprotected. So let's just illustrate a little picture how cover crops affect the soil. And another way that our world is changing in agriculture nowadays is herbicide resistance. We're losing herbicide options. We're having to go back to tillage, and that's a very ancient practice. It still works against those resistant weeds, but our soil is suffering very damage from that. So cover crops, they can reduce soil erosion by simply covering the ground and also suppressing weeds at the same time. So cover crops are a sustainable practice. They regenerate the soil and they also improve soil health in so many ways. That's why we research cover crops. And why did I choose cereal rice specifically to talk about in my project? Well, cereal rice is the most used cover crop in the US, according to SDA. And it's cheap, it's easy to establish, it's easy to terminate, and it's adapted to several environments across several latitudes and climatic conditions in the US. So it can provide several economic and environmental benefits like increasing your organic matter, weed suppression, reduce nitrogen leaching, improves water quality, and all. Again, it's a great tool for integrated weed management. It helps you tremendously to grow a nice cover crop. Great for herbicide resistance management and also a very important tool in organic production. And that's just because it has several potential mechanisms of weed control. So that's what we're trying to study in this project. And just a quick story time for y'all. So rye is a great cover crop because it used to be a wheat. You can probably barely see it in that picture, but there's rye in there with a wheat. It's a very big problem still to this day, but it started off as a wheat in wheat states many years ago when we started doing agriculture. And it avoided weeding by mimicry, so it's very adaptive. And then by the time people realized it was good for food as well, and feed, and all sorts of things, it had already weedy traits. So that's why it's a great cover crop. It's very adaptive, it's competitive, and very easy to establish. Not to worry about it too much. So how exactly does rye control weeds? Well, a lot of people are going to say, well, they produce a whole lot of biomass, it mulches the ground, and the weeds can penetrate this mulch layer, right? Yeah, that's part of the answer. But look at this picture right here. You can see there's not a very good looking cereal rye cover crop on the left. You see a lot of bare spots, a lot of open ground. But why does it have so many less weeds than in the bare plot? Right. So there must be something else going on. And this is what we're trying to find out. So there's some of the mechanisms of weed control that cereal, not just cereal, but cover crops in general can provide. And they're usually divided into competitive and non-competitive. And competitive interactions are just those that compete directly for resources. So it's your competition for nutrients, your water, light, and also for space. And we also put mulching in this category because it just doesn't let the weeds go through it. But there are also non-competitive mechanisms that have been found in literature, such as allelopathy, so just putting down chemicals in that soil that just prevent weed growth. It's just a natural thing. Many plants do that. Reduction in soil temperature is another one. It just delays weed emergence. And cover crops can do that by blocking sunlight and reducing evapotranspiration. And also a new way that people are starting to look at cover crops is a reduction in red to far red ratio. It's just changing light quality. I'm going to talk about it more deeply later in the presentation. So if you don't know much about it, it's okay. We'll look into it later. But these are the ones we're going to cover in this part. Mulching properties, competition for nutrients, water, and reduction in soil temperature, as well as a reduction in red for red. So which one of these play the biggest role or play a role in general? by zero. Right? That's our research question. And this brings us to our objective, which are what's the minimum biomass required for good weed suppression here in Southeast Texas? How does CRI change the soil parameters, more specifically nutrients, water, and temperature? And our last objective is to see how CRI affect the life quality environment in the soil surface, where the weeds germinate, and how all of these things contribute to weed suppression. But to figure that out, we just did a simple split plot experiment starting in 2020 and we terminated spring 21. And it's still ongoing this year, but our data is not fully collected. This experiment is still going. So I'm only going to present data for year one. And so our main plot factor was this forest cedar rate, the price. So we're trying to create a range of biomass and see how different ranges affect all of these things we're trying to figure out. 0, 20, 40, and 80 kilograms per hectare. 80 is like an RCI standard from a few publications, and we just tapped it and then ordered it and then had a bigger control. And the subplot factors were determination timing at 2, 4, and 6 weeks before. And they will be referred from now on as early, middle, and late termination time. So late being 2, early being 6. For data collection, we just took soil samples, before we planted the rye and after we terminated to see how it affected the nutrient dynamics in the soil in each plot. And we calculated the change. We also took biomass samples, which is like one of the most important data points that you can have if you're doing cover crop research. The weed densities, the weed science, so we have to know what's going on with the weeds, their emergence patterns, how big they're getting, and so on. And to monitor the soil, moisture, and water, we had some Hoboling parallel sensors. They continuously monitor it like every 15 minutes wirelessly. The only work we do is put them in. And here's the picture of Jody, our friend, with one of them. That's what they look like. And just sit there all season. And also the red to infrared ratio under the canopy. So we're measuring how much light, red light is being intercepted by the CRI canopy. But we'll get to it later on. Data analysis, we just looked at all of it through stats, ANOVA, and a correlation analysis. And we also checked for normality. Now the fun part, all the data. So here's the biomass that we managed to get last year. Those are very, very rainy years. Y'all remember throwing the field last year, remember a whole lot. So this is probably a little higher than average for this area. But as you can see, the highest we got was about 5,000 kilograms per hectare in the late termination time. And we got about 1,000 for the early termination. One interesting thing that we saw from this data is that seeding rate didn't really affect biomass production very much. Only in the medium termination timing, we saw a little bit less on the lower seeding rate. But it tells us that CRI is really good at tillering and filling out those empty spaces, even at a lower seeding rate. So if for whatever reason, like you had a bad year, a bad establishment, and just a few plants came up, you'd still be okay. Or if you just want to save money and seed, then I guess you'll still be okay. But ground coverage is a whole different thing. You get less ground coverage with less seeding rate. But at the end of the season, your biomass is going to be roughly the same. This is kind of what it looked like. We had this terminator, the late termination that's the green strip. You can kind of see the biomass is not that different across seeding rates. Clearly see the fall plots, but the biomass and ground coverage. It's not too different. Continuing.
I'd believe you that I could see them, but you need to tell me what I'm seeing because I don't.
This is fallow. This is the late termination. I believe this right here is a low seeding rate. This is the 40, there's the middle ground average seeding rate, is the highest. You can definitely see a better ground coverage and the higher seeding rates than the lower, but for biomass production, that didn't change. you can also see a whole lot of weed suppression. Don't really see much in the cover crop plots. And here's weed suppression data. So this is, we just correlated weed density with biomass because we were trying to figure out how biomass, like this range of biomass is created, affected weed suppression. And we identified 3 ranges of biomass. So the low biomass, which is that yellow circle, moderate biomass level, which is that red circle, and the high biomass, which is that green circle. So we kind of found that like if you can get past 2,000 kilograms per hectare, at least in this year, in 2021, then you can get like good weed suppression, especially if it's not your only way of controlling weeds. If you have other ways of keeping your food clear, a good herbicide program, that not just one active ingredient and so on, you can still get by with just maybe about 2,000 to 4,000 kilograms per hectare. And this is what all these numbers and pretty circles translate to. You can see there a no cover plot. There's a whole lot of weeds, a lot of grass, annual grass emergence, a lot of broadleaves. And this picture doesn't show, but there's way more Palmer amaranth in the no cover plots than we had in the cover crop plots. And then if you compare the moderate biomass and the high biomass, there's still a little few more weeds in the moderate biomass levels, but not a whole lot more than high biomass. So if you're lucky enough to get a high biomass, you're good to go. You've got great weed suppression. But if you happen to have moderate biomass levels, you're still okay and you'll still be able to get those weeds. Now, moving on to our second objective, which is soil parameters, how cereal change soil, the soil environment. So this is some results of our soil sampling that we did, which calculated initial and final, and what's the difference. So since we have negative values in there, I couldn't fit like an exponential curve or a logarithm curve, because they're all linear curves. But this is what we see. We see a general trend. It's not a terribly strong relationship, but the higher the biomass, the least the nitrogen. So the more nitrogen was removed from the soil into cereal plant material. And especially if you look at the follow plots down at the 0 line, there's actually a general increase in nitrogen, probably from decomposition from effects in the soil, release from microbial activity. Yeah. That's the general trend is the more biomass, the least nitrogen. And that goes along with a lot of the literature in cover crops published so far. Now, the funny thing was with phosphorus, we saw kind of the opposite response. It's not a terribly strong relationship either. There's this general increase in phosphorus with the higher biomass. So, and we hypothesize that it's due to cerealizing something in the soil that is like pulling phosphorus out of the soil particles, generally in accessible pools. And it's possible an increase in arbuscular mycorrhizal fungi activity that is associated with the rhizomes and scavenging that phosphorus out of those deep holes in the soil. Now for potassium, nothing happens. Right? Later, it's all over the place. And my guess is because there's way too much potassium in our soils down at the farm, and it doesn't really affect where I grow it all. So, this is a gentle reminder of our weed suppression thing. So the higher the biomass, the better the weed suppression. And the higher the biomass, the lower the nitrogen. So our conclusion here is it's just not biomass protection, and it's just not nitrogen depletion. There's a combination of both mechanisms going on for total weed suppression. And our future research is going to try to separate those two from each other and try to isolate this factor and see how each of them on their own play out. Now for soil water and temperature, and that is a crazy graph. Bear with me for a minute. There's two. I just pulled the data from 2 plots in here. So the orange lines are from a no cover plot, so a bare plot. Blue lines are from soil from a high biomass plant. The top two lines are maximum temperature, the bottom two are minimum temperature. And the two gray lines at the bottom represent soil water. In the high biomass plot and then in the middle. But what I want you to see it don't look terribly different in most of this graph, right? The average maximum temperatures for each day. And by the way, this is like March and May, right? When you're terminating the cover crop and you're planting cotton and getting your first wheat emergence. But what I want you to focus on is these two peaks right there. Remember that orange is the no cover. You're getting a much higher temperature peak by the time you're planting your crop. By the time the soil is warming up, there's also a lower minimum temperature peak right there around the same time. And our hypothesis is because rye is actively shielding the soil from sunlight and it's keeping the evaporation low in that sense, so that you don't have as much of temperature amplitude. This has pretty serious implications for wheat emergence patterns. So instead of summer wheat emerging early in the summer, they're going to emerge a little later, hopefully after you plant your cotton. And that way your cotton is going to have a slight competitive advantage, right? And by the time you're going to spray your weeds are going to be a little shorter, so then you're going to be able to kill them a little bit with the chemical herbicide. And hopefully you're going to get your canopy closure before your weeds start producing. And just a quick note about water. Remember this was a very rainy year, so you don't see a whole lot of difference in soil water. But I want to point out, you see there's a little dip in soil water in the cover crop plot, and that's because right around this time is when the rye is starting to flower and put on the most biomass. Remember the biomass graph, how it exponentially increases later in the season? So this is what's happening. But as soon as we have a rainfall event, it just evens out. But this is for a rainy year, and I want to see how it plays out in a more of a like dry year like we had this year. So, but that data will be for another time. Now remember we're talking about red to far red ratio. So if you don't know what that is, it's just how much red light is coming, like how much red light is touching the plant and how much far red light is touching the plant. And that ratio tells the plant whether it should prepare for like a shade competition or not. So far red light is that bit of red light. It's right next to the photosynthetically active range. So the plants don't really use it for photosynthesis. So most of it penetrates through the leaf and reaches the soil, even through a canopy. So if a plant, the red to far red ratio is like plant seeing when there's under competition. So generally, low red to far red causes plants to grow skinny and tall and more shoot allocation, less root allocation, and like less leaf area. Some species don't even germinate if the red to far red ratio is too low. So it's just a way to avoid competition. And what we see is zero rye can increase significantly your red to far red ratio. That's standard. What we see though is like the higher the seeding rate, although it didn't affect the biomass, seeding rate really affected that light interception. So the higher seeding rates in blue right there, you can see there's a general dip in red to far red ratio. The higher the seeding rate. And also the termination timings, the later you let it go for, the more light interception you're gonna have. But we're also doing a greenhouse study to better prove this point, but I don't have time to talk about it today and show you the data, but this is just an illustration of what it looks like. We have dead rye, we have living rye, we have dead rye that was painted green, which is that right there next to the brown rye, to see if the green color is affecting anything. And then we have a bear control. Some of the preliminary results, you can see that Palmer amaranth started flowering a lot earlier than the bear tree, even though they look really pitiful. It's already flowering right there. Look at this terrible looking cover crop. and it still causes like a delay in flowering. So we're gonna study this a lot deeper. We're doing it this year. We're gonna really look into how the rye is affecting like weed development in itself. So a few conclusions. The minimum biomass, at least for 2021, we still have to see how different climatic conditions play in this number. The minimum biomass for weed control is around 2,000 kilograms. It's not terribly high, but it still does a good job of keeping your weeds down. Also, rye can reduce the maximum soil temperature during the warming days in the spring. And that can cause a shift in weed emergence to a later date and give your crop a competitive advantage. And also, rye can induce shade avoidance responses in plants by reducing that red to violet ratio, either by just like not germinating at all, or by like weakening some weeds that try to grow skinnier and taller. And those are more susceptible to herbicide damage. That's some of the literature stated. But the biggest conclusion I take from this project is it's a combination of mechanisms. Not just one, it's just not biomass, it's not competition, it's not allelopathy, but it's a combination of all of them that cause weed suppression by cereal rye. And that's why rye is such a great weed suppression. So this early spring, you see horseweed growing like crazy in the fallow, and annual grass is all popping up, and not a whole lot of weed pressure in the rye plants. And regardless of mechanism, cereal rye is still a great tool for integrated weed management. Now, though cover crops are not the answer to all our problems in agriculture, it is part of the answer. So I would like to tell that to people, and hopefully this will be studied more deeply and it'll be a practice more widely adapted overall in the US and in the world as well. And I'd like to thank you everybody for listening to me, and thank you for all of you people sitting here and online for your help and support of everything. And I'll take any questions.
Did you take any data on the cotton yield or production after your cereal rye was terminated?
No, we do not take any agronomic data in cotton because it's more of a purely ecological ecology project. It's an interaction between weed and rye. The cotton was just there to like simulate the area. But there's a lot of studies showing that interactions between cotton and cover crops out there. We're trying to focus more on the weed side.
I had the seminar on in my office before I came down, and the speakers going in and out. I'm assuming every time he walks away from the film mic, he goes out.
Okay, that's probably a result of, we got another mic in the room and
I heard David clearly.
they rewired
Couldn't hear
things.
the speaker.
Okay, thanks very much. So you'll turn that around.
No, you can hear the audience, can't hear you.
Okay.
When you were talking about nitrogen and phosphorus, was that total nitrogen and total phosphorus, or was that only inorganic nitrogen and exchangeable phosphorus?
Whatever comes out in the soil test, so that's I think that's nitrate and phosphorus, phosphorus that's in the solution. So,
Can you go back to your nitrogen results? So I've got a few questions, but just in the interest of time here. So you said that so I think your X and Y axes are backwards, right? Because you're trying to prove the change in, is that right?
yes, so the bottom, the x-axis is the change in nitrogen. So to the left is negative because it's changing.
Right, but you're trying to show the biomass is changing nitrogen. So your axes should probably be flipped, but on the low biomass, you said that that's fallow, right?
Yes, so the ones on the zero are fat.
So that's not really part of the same experiment in a way. It's not the same condition. So if you take those out, I don't think you actually have a correlation there, right?
Yeah, it's a weak correlation, but
I mean, that correlation is mainly driven by all those fallow plots there, I think.
yeah, because the fallow doesn't have the cover crop, so it doesn't have to take the nitrogen at all. So that's what we're trying to prove, like a no cover crop versus cover crop scenario, not as much as the biomass, only biomass.
Well, the cover crop was- If it's even fallow, the weeds will still take up nitrogen, right?
I mean, that's a qualitative variable, whether it's fallow or not. But what you were saying here was that the amount of biomass changes the amount of nitrogen, which is a quantitative and sort of a separate analysis. And by pooling both those treatments, you're sort of getting a driver of the fallow experiment here. Whereas it doesn't really address the, as the biomass quantitatively changes, does it affect the nitrogen? So you almost have to pull out those fallow treatments, I think.
Yeah, that's a good point. Maybe I might suggest, we need to normalize the error and factor value from. And
So I guess the biomass didn't have as much effect on the nitrogen on its own, but is that more of a, it is cover crop or no cover crop? So, but thanks for pointing this out.
So it took me a little while to tune in. And so I might have missed it, but then again, I might have heard you. I think you said that rye was a weed.
Used to be, and now it still is in some places.
But rye has been a crop since civilization.
Mhm.
I mean, it's also been a weed because a plant out of place is a weed.
Yeah,
But I'm struggling a little with its origin and its development as a cereal crop was very much as a grain crop.
so there are different theories, but one of the theories that I looked at the most, it was originated around Turkey and Ukraine, that region of the world.
Yeah.
And when people start farming wheat, it just got into the crops. And then it kind of developed with the wheat as a weed for a while through mimicry, and it looks very similar even to this day. They look very similar when they're young.
Which
Very hard to
rye did you use?
variety. It's called. I can tell you.
Was it Elbon?
No,
Okay.
sorry, I'll
Because
owe you that one.
we'll talk later.
Let's thank our speaker again.